Hello everyone, in this video I'm going to show you how to use Proteus2, an open access web server that helps predict the secondary and tertiary structure of protein sequences. I will also show you how you can view the results obtained from a Proteus2 prediction using an online structure viewer. As you may know, there are many secondary structure prediction web servers online such as Cypred, JNet, and Gore that do an excellent job of predicting protein secondary structures. However, unlike other web servers, Proteus2 also identifies signal peptides, predicts the location of membrane-spanning helices, identifies membrane-spanning beta strands, and performs 3D structure prediction via homology modeling. But what really makes Proteus2 stand out is its high prediction accuracy. When compared to other web servers, Proteus2 achieves the highest prediction accuracy as measured by the Q3 black and SOV gray scores shown in this bar chart here. If you're interested, you can find more details regarding head-to-head comparisons with different programs and web servers in the Proteus2 website under the Documentations tab. Now, let's get started with our tutorial on how to use Proteus2. Open your internet browser and go to Google. In the search bar, type Proteus2. Next, click on the link that reads www.proteus2.ca. For your convenience, I have attached a link in the description box. When opening the website, you'll see that there are 4 different tabs: Home, Documentation, Sample Output, and Contact and Download. The Home tab is Proteus2's home page, and this is where you'll be submitting your FASTA protein sequence for secondary and/or tertiary structure prediction. Under the Documentation tab, you'll find more information about how to use Proteus2 and additional details on how it works. Under the Sample Output tab, you'll find examples of the different kinds of Proteus2 outputs. And finally, under the Contact and Download tab, you will find programmer contact information, a link to the source code, and, if you're interested, instructions on how to install a local version of this service on your OS. Now, let's go back to the Home tab. Be sure to have a protein sequence in FASTA format ready. For this tutorial, I prepared one right here, and if you'd like to follow along, I provided a link to download the same file that I'm using for this example in the description box. Now that you have your FASTA protein sequence, you're ready to submit it to the Proteus2 web server. First, make sure that you check the Run Secondary Structure Prediction box and select the organism type. For this example, our FASTA sequence belongs to a eukaryote. It is important to note that there are several different options for submitting sequences to Proteus2. For instance, we may copy and paste our single sequence right here in this box, or choose to upload our sequence file here. In addition, we have the option of viewing our results in the web browser, or we can have them emailed to us instead. For this example, I'm going to copy and paste my sequence here. I'm also going to select the 3D PDB homology modeling option, as we're going to ask Proteus2 to perform homology modeling as well as secondary structure prediction. When you're satisfied with all your selected options, click on the Submit button. Clicking Submit will redirect us to another page that assigns our query a unique ID number. The Proteus2 program is now processing our sequence. This page will automatically refresh every few seconds as Proteus2 runs a series of tests and updates us on its progress. An average Proteus2 run should only take about 2 minutes to complete. I'll get back to this once it's done. Now that it's done, the page now displays our prediction results. The summary section shows submission statistics such as the time the sequence was submitted and how many residues were read. It also indicates if the query sequence contained a transmembrane helical region or a signal peptide, the number of homologs found, the number of sequence alignments, the overall confidence value of the predictions, and the predicted secondary structure content. Scrolling down the page, we are also given a nice graphic detailing the sequence alignment of the homologs and the final predicted secondary structure. Scrolling down further, we can find the predicted signal peptide of transmembrane structures. For our particular example, there are no signal peptide or transmembrane structures. Below these graphical images is a section called Detailed Prediction Information, highlighting the various tools employed such as SciPred and JNet that were used to generate this secondary structure prediction. Finally, at the bottom of the page in the homology modeling section is the predicted 3D structure of our query sequence, which is now contained in a PDB file. We can view a 3D rendering of our query sequence using the built-in web model viewer here. In summary, we learned how to navigate in the Proteus2 website, submit a FASTA formatted protein sequence, customize our output by changing some options, interpret the results, and finally, view our results in a PDB viewer. I hope you enjoyed this tutorial, and please stay tuned for more videos to come.